Hello everyone, we are Crystallinica. We're a team of engineers and physicists on a mission to empower researchers and drug developers to pioneer treatments for neurological and cardiac disorders. On the path from the lab bench to the pharmacy counter, all drugs undergo an extensive screening process. This can take anywhere from 10 to 15 years and cost in excess of $3 billion US. If a new drug candidate is found to fail a test at any stage along this path, it gets scrapped, and all the money and time that's been spent on it up to that point is lost. As part of the preclinical screening phase, where cost savings are maximized in the event of a failed candidate, new drugs are screened for their potential side effects on the brain and heart by looking at the impact of the drug on the cells of these electrically active organs. Historically, this was done with a technology called a patch clamp, which was so revolutionary it won its inventors the Nobel Prize in Physiology in 1991. Since then, the technology has scaled to something called a multi-electrode array, which enables recording from anywhere from 10 to upwards of 64,000 individual sites. But even the world's leading-edge multi-electrode arrays are still undersampling the true complexity of the dynamics of these biological networks. We're introducing a fundamentally new technology called the diamond optrode array, and this enables more than 10 times the resolution of the world's leading-edge MEAs. And we liken this to going from a 0.06 megapixel camera up to a greater than 4 megapixel camera. So before the TRAM track program, our hypothesis was quite simply, when it comes to electrodes, more and closer together is better. And in attempting to test this hypothesis, we were exposed to the whole ecosystem of MEA end users. And what we learned through our interview process was that, yes, in principle, more electrodes is always better, but broad industry adoption does require someone to do the work to validate to the industry exactly how more electrodes are better. And what this essentially means is that our first market is academic researchers. So continuing our ongoing collaborative research projects, our next step is to get the technology into the hands of independent neuroscience and cardiac research labs. From there, as we scale the system to enable higher throughputs, the next group of uptakers will be pharmacology labs. And it's through them that we'll be introduced to industrial R&D labs, and that is what's going to pave the way then for industry adoption in the in vitro toxicology screening sector. So the next steps for us, I'll start by saying that none of this would be possible without the seed funding and access to the TRAM program enabled by the Faculty of Science E=mc² program. So a very big thank you to the Faculty of Science for that. Hopefully we will be participating in the next series of TRAM programs, and we'll be moving to secure our intellectual property as soon as possible. From there, to guide us through the commercialization pathway, we'll need to establish an advisory board. So if anyone in the audience has relevant expertise or interest in getting involved with the project, we would really be interested to hear from you. So once again, we are Crystallinica. We're developing a new type of electrophysiology platform with serious potential for use in the drug development pathway. Thank you very much for listening.